If you look closely at the tissues making up the human body, the physical variation is staggering. Consider a flat, tightly packed skin cell meant to form a barrier, compared to a brain neuron stretching out a vast network of delicate connections. The skin cell is constantly regenerating and producing keratin to armor the body. The neuron, meanwhile, generates electrical impulses and builds complex synapses to process information. Yet inside the nucleus of both of these radically different cells, you will find the exact same inventory, 46 chromosomes. The underlying genetic code, every single base pair of DNA, is a 100% match. It is tempting to think of the DNA sequence as the sole architect of the cell, but a matching blueprint cannot explain why these two buildings look nothing alike. If the genetic code is identical, what physical system forces the skin cell and the neuron to execute that code so differently? The physical architecture of the genome governs how these identical instructions are interpreted. DNA is not a static string of text floating freely in the cell. It is a highly dynamic, physical landscape that constantly shifts shapes and states. To understand how cell identity is mechanically controlled, we have to look at the structural hierarchy of DNA, zooming in from the macro-level chromosome all the way down to individual molecular tags. What dictates cellular identity is ultimately a mechanical problem of physical access. It depends entirely on how tightly the genetic library is packed. We begin our descent at the absolute highest level of DNA packaging. This is the chromosome, the peak state of macrocompaction. Human cells contain precisely 46 of these distinct, highly condensed structures. Think of a chromosome as the ultimate storage crate. A cell only packs its DNA into these tight X shapes when it's actively preparing to divide. This extreme coiling serves a strictly mechanical purpose. It ensures the massive genetic strands can be split evenly between two new cells without tangling. These packages are so dense that they represent the only state where DNA is actually visible under a standard light microscope. While this compacted state is highly secure for transport, the extreme density makes reading the individual genes physically impossible. To actually use its DNA and establish its identity, the cell must unpack these storage crates into a form that can be read. Once a cell finishes dividing, it enters its normal life cycle. During this phase, the DNA exists as chromatin, the functional, uncoiled state of the genome. This is the cell's open library, the structural phase where the genetic code is actually accessible. To maintain its shape, chromatin consists of DNA wrapped around a repeating physical scaffolding of structural proteins. This complex matrix dynamically shifts between two distinct states, adapting to exactly what the cell needs at any given moment. The looser state is called euchromatin. Here, the DNA is active, with wide enough gaps for molecular machinery to slip in and read the code. The tightly packed state is called heterochromatin. This DNA is silent, inaccessible to the readers, heavily knotted into impenetrable clusters. The balance between these active and silent states is not fixed, it constantly fluctuates in real time. By controlling the regional density of chromatin, the cell dictates exactly which sections of its genetic library are open to the public and which remain firmly locked away.
Precise coiling and uncoiling at this scale relies on a foundational mechanical unit. This is the nucleosome, a structure often described by biologists as beads on a string. The nucleosome is the fundamental structural unit of all chromatin. If the DNA is a continuous thread, the nucleosome is the individual spool holding that thread in place. Looking at the physical geometry, a distinct segment of DNA wraps roughly 1.65 times around this central core. The core itself is constructed of exactly 8 interlocking histone proteins. This primary structure offers an immediate mathematical advantage for space management. This initial wrapping step alone compacts the massive, sprawling length of the DNA strand by a factor of 6. Without these histone spools acting as foundational anchors, the cell would have no mechanical way to organize or contain its immense genetic sequence. But how does a cell coordinate millions of these individual spools to create the loose or dense chromatin fields we saw earlier? This is managed through a mechanism known as epigenetic regulation. Epigenetic regulation refers to specific chemical modifications made directly to the DNA or the histone proteins without changing the underlying sequence of the genetic code. You can think of these modifications as molecular sticky notes, tags that instruct the spool to either open up or clamp shut. One major type of tag is the methyl group. When enzymes attach methyl groups to the histones, it typically locks the chromatin tight, packing the nucleosomes together and mechanically silencing the enclosed genes. A contrasting tag is the acetyl group. Adding acetyl groups forces the nucleosomes to repel each other. This loosens the histones and grants access to the genes within that gap. This push and pull dynamic acts as a molecular volume dial, smoothly altering the packing density of the chromatin across the nucleus. Ultimately, these epigenetic tags serve as the mechanical authority on whether a sequence is completely active or effectively invisible. All of this structural packing and unpacking leads to one biological outcome: gene expression. Gene expression is the process where a specific DNA sequence is read, converted into RNA, and used to build a functional protein. This is the actual execution of the genetic instructions. But there is a strict mechanical prerequisite for this machinery to function. The transcription machinery can only bind to the DNA and produce RNA if epigenetic tags, like acetyl groups, have successfully loosened the nucleosomes in that specific region. If those same nucleosomes are methylated instead, the region is compressed. When the local chromatin is locked tight, the bulky transcription machinery literally bounces off the exterior. The gene remains unread and unexpressed. This brings us back to the opening paradox of the skin cell and the brain cell. Both possess identical sequences of DNA, but they carry completely different epigenetic maps. The genes responsible for firing neural networks are acetylated and held open in the brain cell, while those exact same genes are heavily methylated and locked shut in the skin cell. Ultimately, the physical machinery of the cell determines identity by controlling which chapters of the library remain open and which are effectively locked away.